Hello, everyone. Thank you for participating in today's Zoom meeting on the development of a gene therapy for Pompe disease, recent history. Today's guest speaker is Dr. Dwight Koeberl. Dr. Koeberl joined the Division of Medical Genetics and Pediatrics at Duke University in 1999. His group has developed new gene therapy and genome editing for glycogen storage diseases, including von Gierke disease and Pompe disease. In January 2019, the division initiated a clinical trial of gene therapy for Pompe disease. As Dr. Koeberl goes through his presentation, you can send questions at any time by clicking the Q&A feature at the bottom of the Zoom window. Dr. Koeberl will answer your questions at the end of the presentation. Thank you very much, and with that, I will pass it over to Dr. Koeberl.
Hi, everyone. Can everyone see the slides? And hear me?
Yes.
Good,
I can see
great.
the slides.
Yeah, thanks for this opportunity to present our research. First off, just some disclosures, and mainly the top and the bottom bullets here. It's possible that Duke University and myself could benefit financially if the treatments talked about here prove effective and are successfully developed commercially. Also, I've had equity in AskBio, the sponsor previously. I know that most of you are familiar, but just a brief background on AAV vector-mediated gene therapy. A vector is a modified virus like an AAV. It's a gene delivery vehicle. It carries something called a transgene to make a protein like GAA to treat Pompe disease. And so this little cartoon in the lower right shows that a transgene has these parts, promoter, cDNA, which codes for the protein polyadenylation. And it's packaged in this AAV vector genome, just a little of the viral sequence at each end. And then at the end, it's an AAV particle, so viral particle, and that's why it can be taken up into the tissues, taking advantage of the AAV life cycle. So this is an example of two approaches to gene therapy, either infusion of a vector like an IV medication, or injecting a treated stem cell. And we and others are proceeding with the infusion of the vector, like shown on the left here, with a simple IV. For Pompe disease, as this audience knows, there's a deficiency of GAA that leads to accumulations of glycogen in the lysosomes in muscle. This relates in, it causes muscle weakness, hypotonia, in some patients a cardiomyopathy, and can result in cardiorespiratory failure. The treatment that has been developed by Dr. Chen at Duke and other laboratories what is an enzyme replacement therapy, and this takes advantage of the possibility of injecting the GAA into the bloodstream where it can bind a receptor on muscle cells and then be trafficked to the lysosomes where it's activated and will degrade the accumulated glycogen. We've developed this as a gene therapy. It's intended as a one-time gene replacement therapy that will make GAA continuously. In our strategy, actually, this is an AAV8, unique kind of AAV that goes to the liver very efficiently. Also, the promoter produces GAA only in liver. It's liver specific. And at least in our experiments, there are a couple of advantages. That liver specific expression suppresses antibody responses against the GAA that can interfere with benefits from GAA replacement. And also the GAA is secreted from the liver, it flows to the blood, and is taken up in the heart and skeletal muscle. And again, in our experiments in the laboratory, it can improve muscle function. This is an example of one of those early experiments. One of the scientists in the laboratory made the vector. We administered it to GAA knockout mice with Pompe disease, and you can see the GAA in the blood, very stable and increasing out to 12 weeks. And it compares with the GAA protein over here on the right, good quantity, the same size. And then when we treated our mice, you could see a Pompe disease mouse heart in the middle with high amounts of purple stained glycogen. And then on the right, that is cleared from the mouse treated with gene therapy at 12 weeks. And it looks very much like the wild type or normal mouse heart in the middle. Some advantages of this approach to gene therapy then, you could see that with regard to the GAA protein in the blood, its activity is clearly elevated for mice treated with gene therapy. Now we're comparing to ERT, and it's sometime after the last dose of ERT, but we know for ERT there's a large amount initially of the GAA protein in blood, and then it's rapidly cleared. And so at this time point, you can see that the AAV is expressing continuously, and the GAA from ERT is gone in the blood. And then showing the immune tolerance induction or the prevention of antibodies. In the middle, mice treated with ERT have antibodies. On the right, mice treated with gene therapy don't. Now here, this is more about that study. So it was a low dose gene therapy, just one treatment at the beginning, and then 4 weekly doses of ERT. We stopped here because actually the mice developed antibodies against ERT, and then they have a hypersensitivity or allergic response to subsequent doses. So without modifying the mouse to prevent this allergic response, we can't go beyond about a month. But you can see here the GAA is coming up in the liver, a little bit more for gene therapy as compared to ERT a week after the last dose. And then importantly, the glycogen is coming down, and the glycogen is more sensitive for detecting the biochemical correction of Pompe disease. So both for ERT in the gray, and then the very low bar here for AAV gene therapy, the glycogen is really decreased markedly. And that's 5 weeks after the initial treatment. Similarly, for the diaphragm, we're seeing a nice reduction of glycogen from this either ERT or low-dose gene therapy. And for the quad, actually, if we look only at males, we could see an improvement in the glycogen in the quadriceps. Again, from a fairly low-dose gene therapy that, based on this study, could potentially replace ERT as a treatment. Potential advantages then for the gene therapy, again, based on our laboratory experiments or preclinical data, the continuous GAA produced in the bloodstream, increased delivery to muscle, immune tolerance induction, potentially treating a larger patient population because it would be a one-time treatment that didn't require repeat visits and lots of support to be able to do that, and potentially a more complete correction. So just a minute on variations then. So there's something that's been developed in gene therapy called Secretable GAA. And we did a study of this in 2006. It's modifying a part of the GAA protein so it escapes the liver more readily and then could be present in the blood at a higher level. So this SP version is the secretable version. And you can see it's higher in the blood compared with regular GAA on the left. Then when we looked at the tissues, for instance, the liver, where it's being made for regular GAA, unmodified, there's quite a lot in the liver. And then for the secretable form, there's less in the liver, more of it's getting into the blood. And what did this result in for the tissues? And this is just all secretable GAA, but you can see that it gets into heart, diaphragm, quadriceps, even the brain to some extent. And then the upper form is the form that's actually secreted, taken up from the blood. The lower form is fully activated and would be decreasing the glycogen there. So it seems to be working well. And this concept has been developed extensively. This publication a few years back, about 5 years ago, from the group at Genethon, and a couple of Duke researchers did contribute to this as well. But the idea was making a secretable GAA, testing it, Now, one kind of interesting feature of this is some of the modifications of the GAA DNA seem to be associated with more antibody formation. But then when the protein was modified to make it secretable, those antibodies went away. And then down here on the bottom, sorry, it's so small, we can see that for a higher dose, there's a very good reduction of glycogen in the muscle compared with the red bar for all these different vectors. And at 10 months, it's even a greater correction. And so this strategy has been developed by the Spark group further, and that's ongoing research and a clinical trial. This is some more data from Jonathan talking about now long-term, how does gene therapy do compared with ERT? And these mice were modified so they didn't make antibodies against the GAA, and this long-term experiment was possible. The mice didn't have the allergic response. And the glycogen content of the heart responds really nicely to replacement therapy, showed that the ERT reduced it nicely, as well as a variety of doses of gene therapy down to a pretty low dose. And so this, reinforces the concept, this is layering on one graph ERT with really high GAA after each infusion that drops down. And this is based on real data, how the GAA goes extremely high after ERT and drops down in the following days, compared with the level of GAA that gene therapy produces steadily. And you can see from the previous slide that even a low level of GAA, lower than for ERT, but continuous, really can treat Pompe disease. So this is the concept behind this liver-based gene therapy for Pompe disease. And then this is another experiment from our group, again, a moderate dose comparable to what I showed you for the secretable GAA, but out past 6 months and really reducing the glycogen content compared with untreated mice with Pompe disease across the heart, diaphragm, quadriceps, and other muscles. And the mice actually had improvement on this rotarod testing that measures their neuromuscular function. So we could call it a clinical benefit. So either a normal unmodified GAA or a secretable GAA can be used for a liver-based gene therapy. So moving ahead then to the clinical trial, and this is based in Duke, a single site. It was supported by 2 institutes at NIH, and then the support switched over to AskBio, our current sponsor. It was a typical design for a phase 1 clinical trial where the main concern is safety. 2 groups of 3 participants each for a medium and then a higher dose. And this did feature prednisone immune suppression from the beginning. More on that in a second. Safety is the main endpoint. So at multiple visits, we monitored what was going on in terms of symptoms, any adverse events, as well as laboratory testing, liver enzymes, muscle enzymes, to monitor safety. And then secondary endpoints were more for efficacy or benefits, and some that are fairly unique, but that we developed, for our mouse experiments, the GAA activity in the blood or in the muscle. We also looked at antibodies against GAA that we didn't expect to see, we didn't. And then we measured muscle function, 6-minute walk test, the forced vital capacity from pulmonary function testing to monitor how the patients were doing clinically. Then a little bit more about the immune responses, what this means, and the need for immune suppression with prednisone. So we know that above a certain dose of AAV, actually in clinical trials, there is a T cell response, so what are called cytotoxic T cells, actually against the cells that have the AAV, and that's going to eliminate the AAV entirely and eliminate any benefit from gene therapy. So prednisone, given either at the time the T cells appear or to prevent the T cells appearing, can save the gene therapy. And here is the timeline. for the design of the clinical trial. We started at enrollment, monitoring key criteria to select patients who would be eligible according to the protocol. Things like a clear diagnosis of Pompe disease, having been treated for over 2 years with ERT at the same dose to be sure there's no continuing benefit from ERT. Any new benefit would be from gene therapy, being adults, and not having any antibodies against the AAV that could prevent it from working. During the clinical trial, at these multiple visits over 52 weeks, measuring the GAA in blood, like liver enzymes, looking for the T cell response, periodically doing a muscle biopsy both before and after gene therapy, muscle function testing like the 6-minute walk test, the pulmonary function tests. And a key feature here was the steroid or prednisone treatment that started 1 day before the vector, continued for 4 weeks at a full dose, and then there was a gradual taper going down in the dose weekly out to week 15 when it stopped. We monitored the patients carefully with a special attention at week 24, when by certain criteria we could stop the next ERT dose at week 26, stop ERT, and then monitor for the rest of the year out to week 52. These are the kind of adverse events that we both measured and patients reported. And the ones that were related to the study drug or gene therapy are the most important. They're all important, but we need to understand, what is the gene therapy doing? And what was observed for these first 3 participants were things that were expected and were graded as mild to moderate, and then resolved, like headache, muscle ache, and then some slight increase in the liver enzymes. That can signal a T cell response against the AAV, but actually this was explained by another illness. So we think that the immune suppression was successful at preventing any T cell response against the AAV for this first cohort. Other serious adverse events were not related to the study drug, but they deserve special attention. And it wasn't my determination that they weren't related to study drug. There was an independent clinical investigator who made these decisions. So things, though, that could be explained by having a chronic disease like Pompe disease and were not related to the gene therapy. With regard to stopping ERT at week 26, all 3 participants met the criteria for that included elevated GAA in the blood compared to at baseline before gene therapy, and then no decline in terms of muscle function testing. And we measured in the blood GAA that was at least a little bit increased compared with the start before gene therapy at week 52. The muscle GAA was significantly increased with a p-value of less than 0.05 at week 52 compared to screening or baseline before gene therapy. The muscle glycogen content seemed to go up a little bit for 2 participants at week 52. So this might be telling us that we need a somewhat higher dose of gene therapy to effectively replace ERT and treat Pompe disease long-term. And then there's a urine biomarker that's basically stayed the same over the course of the study. So the conclusions were, based on the assessment of related adverse events, the lack of related serious adverse events, that there was initial safety with regard to bioactivity or whether the vector was working. Especially, we saw a significant elevation of GAA in muscle at week 52. And then there were the criteria for stopping ERT were met after week 24, and just that the patients stopped and remained off ERT out for a total of about 18 months for this group. And we published this in the past year in Molecular Therapy. You could look at that publication for more details. I wanted to update the audience about ongoing clinical trials based on information that's publicly available. So for Audentes or Astellas, now Astellas, they have an ongoing clinical trial that is more based on treating the muscle as opposed to a liver basis. Spark Therapeutics has a study that's more liver-based. And then I've been talking about this Glypios biopharmaceutical study, or AskBio. With regard to Astellas, I have a little information from abstracts presented at the World Meeting 22 and 23. So their vector, AT845, is an AV8 that's given in a high enough dose and is designed to express GAA in muscle. And initially, they talked about 4 participants, 2 at a lower dose. It's actually about 20-fold higher than the dose I talked about in the liver-based study, the one that we published, but it's higher dose to get to muscle. And then 2 at a 2-fold increased dose compared with the starting dose. And in that initial abstract, they mentioned 1 patient having an elevated liver enzyme, ALT, that signified T-cell response, and that was treated with prednisone and suppressed. And they mentioned that they could detect the vector DNA in the muscle, so clearly it was getting there to the target. In 23, a brief update, and 1 E14 vector genomes per kilogram, and higher dose was added, again, with the goal of really delivering the gene to the muscle. All patients were treated with prednisone, knowing that the T-cell response will happen at this kind of dose. And they did have a serious adverse event that was related, a peripheral sensory neuropathy, and were placed on clinical hold by the FDA, but that has been addressed, and they've reopened the clinical trial. With regard to Spark, I actually don't have information, but I really like this quote from their website. It sounds strange to say, but it's a really exciting time. We have so many wonderful people and companies putting their efforts and talents into helping people with rare diseases, and that's really inspiring. Being involved with this and hearing about all the work that's being done in the field. We know that Spark has this ongoing clinical trial. You heard about it in a previous AMDA webcast, but I don't have any data that have been released publicly. And I can't share that today. And then this is just the AskBio website. You could see more about the clinical trial that I've presented. So I'm going to switch gears here a little bit and try to share some thoughts about being in a clinical trial, important background information. So we really need to acknowledge that genetic therapies like gene therapies are novel. They've just been developed. There's not a lot that's known about all the possible effects. So these risks remain unknown. We know generally that they're dose related, so a higher dose is a greater risk, and somewhat that they're preventable, pardon me, depending on the design of the study, maybe the dose, and also that they're treatable. So some of the serious adverse events, or SAEs, have actually been even life-threatening or fatal. There have been a couple of patient deaths. One was in a clinical trial for Duchenne muscular dystrophy. The second was following the FDA approval of a gene therapy for SMA. More on that with the next slide. Just a principle here, it's really important that these events be shared with the community. So they must be reported to the FDA, and the FDA has to evaluate them and respond. And I certainly trust the FDA to do that. But I think it's also important to share the knowledge, share the information about what's happening in clinical trials. Now, a good example of that is in the field of gene therapy for Duchenne muscular dystrophy, including this patient death that was mentioned from the Pfizer clinical trial. There was a meeting convened by a patient group called the Parent Project, along with industry and NIH, where these SAEs were discussed openly. There was sharing between the industry sponsors and with NIH, and that was a really important effort, and that should be replicated, I'd say, for gene therapy generally. Long-term follow-up studies are really, really important. That's illustrated by SMA, where really the side effects or adverse events became much more apparent following FDA approval, when many, many patients were treated, and actually, those patients are followed, and those adverse events are reported to FDA as after, in post-approval monitoring or phase 4 research. And then there are unknown risks that remain that have never been seen in patients, or at least there's only a hint that they might happen. And so the bottom panel here is a paper that was published in animals that were treated at extremely high doses with a vector that is like the one that's approved for SMA. And they developed brain involvement with the basal ganglia showing toxicity. Actually, the basal ganglia control movement. And so these animals developed a neurologic complication that hasn't been seen in humans, but there's a potential risk for that. This is just emphasizing that ongoing research is really important, and that there are unknown risks. We learn about these from clinical trials, but really the laboratory research is so important in terms of predicting them. So more than about the most serious SAEs from AV gene therapy. And they involved typically almost entirely what's termed TMA for thrombotic microangiopathy. This involves activation of complement, which is a special arm of the immune system. It usually responds very quickly to, say, a bacterial infection where there are antibodies against the bacteria and the complement is activated. But this can happen in response to a drug. And there's strong evidence saying this happens in response to very high doses of AAV vectors. And the kind of the three effects from complement activation include tissue damage, like kidney or heart. There's been kidney shutdown. For the Duchenne patient, there was evidence that the heart was inflamed and damaged. We can see changes in the blood counts, low platelets. That's the thrombotic part. We can see hemolytic anemia, so breakdown of the red blood cells, and they look quite unique under the microscope. So all the signs now of TMA from AAV gene therapy are recognized. There have been 2 documented patient deaths from this. One, again, for a patient treated with approved SMA gene therapy. It is rare. Hundreds of patients have been treated with that gene therapy, and one in a clinical trial for Duchenne muscular dystrophy. And of note, these doses are 50 to 100 times higher than for the low-dose liver-targeted gene therapy that I've been mentioning. And in general, there seems to be a slight advantage for dosing with liver as a target, avoiding those really high doses of AAV. But multiple strategies are important for trying to develop gene therapy, and this is just one factor in thinking about those. But if we now think about risks versus benefits, what are they? So liver toxicity that I've mentioned, at higher doses of gene therapy, we know that it's not even preventable, that it can only be treated with immune suppression. And this is known from, say, SMA gene therapy. But generally, it's not an issue so much. Patients typically remain asymptomatic. And the real risk is that for a liver-based gene therapy, the benefits will be lost. And again, this has to do with the T cell response, the immune response, specifically to AAV in the liver. Complement activation, like I showed in the previous slide, causing the TMA, is severe effects, almost an immediate or very acute effect, kidney failure, low platelets, and requires specialized drug therapy just to manage it. And we know it can actually cause mortality. there's risk from immune suppression. So say being routinely dosed with prednisone to be in a clinical trial, there are risks of, having an infection. Usually this doesn't happen, but it's a known risk. And certainly other side effects from these steroid medications can disturb sleep, and they have other effects. So really minimizing the immune suppression or adapting it to be ideal is important. And then there are long-term risks, both hypothetical from animal experiments and unknown that we haven't even seen yet. And these can potentially be serious. So, maybe I've emphasized too much the risk, but we have to acknowledge them versus potential benefits. So specifically for Pompe disease, we could view a treatment that replaces ERT as a potential benefit. I think that's an idea that has to be further developed. I don't think it's something that FDA is accepting as benefit, as a proven benefit. But stability of symptoms. So it's known that for ERT, there can be a gradual progression of symptoms, say decreased muscle function, not being able to walk as far, maybe decreased pulmonary function, more difficulty with breathing, despite ERT in the years of treatment. And then potentially a gene therapy could actually treat Pompe disease more effectively than an ERT. And this concept is supported by those experiments that showed that a continuous but lower level GAA production is effective at treating Pompe disease and replacing ERT, and perhaps even treating Pompe disease more effectively than ERT in those mouse experiments. So one more thing I want to mention, and this is a new direction we're going in our research, which is just the need to treat early in life and kind of the challenges there. So an experiment we did was to treat our mice with Pompe disease early as infants or later as adults. And we gave them the same number of AAV vector particles, or the same dose, which is what I'm showing here with the blue dots overlying the liver. Now the mouse liver, the infant mouse, is going to grow substantially over their lifetime. But if all this AAV was taken up efficiently, so there are many more AAV particles in the small liver, as it grew, it should end up at the same place as treating an adult, where there were fewer particles per cell. But in our experiment, a year-long experiment, we found this wasn't the case, that actually the infant liver didn't take up and process the AAV really, really efficiently and have the same amount at the end as for the adult treated liver. There was less left. And in fact, the infant mice seem to be clearing the AAV actively from their liver early in life. Now, to address that limitation, knowing that we need to treat patients with infantile onset Pompe disease as early as possible, we are working on a genome editing strategy. So in this experiment, a CRISPR would cut the chromosome at the GAA gene, actually. And then at this double-stranded break, an AAV that we call the donor could pair with the DNA, because there's homology, or the same DNA is present both in the donor and in the chromosome, in the GAA gene. And there would be crossover events, what's called homologous recombination, to integrate that gene, that transgene, into the GAA gene, and then the donor would make GAA. And this is a safe approach. There are risks. Making this double-stranded break could disable a gene, but the GAA gene is already mutant in Pompe disease, so it seems like a reasonably safe strategy. And then all the daughters would make GAA. All the cells descending from this original cell that was treated with genome editing would continue to make GAA. And this contrasts with the situation where AAV is gradually diluted out as cells divide, because it doesn't integrate in the chromosomes. So we're working on this actively. We have NIH funding and hope to be able to tell you about the results in coming months. We have done this and published a study actually in the dog model for von Gierke disease, the GSD1A. On this little cartoon that was published along with our article shows how, for the genome editing cells, kind of clever symbol here, a pencil editing the chromosome in those cells. Those cells stick around; they're very stable, and we monitored from 4 months out to 16 months, and the genome editing cells were exactly at the same level. Whereas cells that just took up the AAV but didn't integrate it, didn't have genome editing, just regular gene therapy, gradually lost the AAV and lost the benefits, didn't make the protein anymore. So this is a concept of treating early in life with a stable effect with genome editing. So that's all I have. I really want to acknowledge my colleagues in medical genetics who teamed up to do all this research, especially Eddie Smith, who's a pediatric neurologist who helped us with the clinical trial. It's great that he was independent from medical genetics and was willing to help us. He's really an expert in terms of clinical trials. Also funding from NIH and our industry partner, AskBio, and also for patients and their families and for participating in this research. And also thanks for your attention. I'll stop there. Any questions? Chime in, don't be shy. Chat, maybe. Okay. Nothing yet. Q&A, yes. Interested to hear Dr. Koeberl's thoughts on considerations beyond the vector and capsid to the sequence used in the cassette. Stability in the bloodstream of the enzyme produced and its cellular uptake mechanism. Yes, and this is a question from Ryan that's really, really detailed. So this is an important consideration, and it's asking about, what about the coding sequence for the GAA? And as I mentioned, it can be modified to make it secretable. It can be wild type. Essentially, the GAA that we used in the clinical trial that we published was a wild type GAA that's very, very similar to the DNA that's used to make myozyme or lumozyme. And that works well. So, one of the other questions then is, how is the GAA produced, and is it properly glycosylated so it binds the receptor and is taken up? And it's well known now that for myozyme, lumozyme, that glycosylation was far from perfect, and it would circulate in blood and not be taken up very efficiently by the receptor and not taken up into muscle cells. So we haven't done the mass spectrometry on GAA from gene therapy in a mouse, but that would be fascinating, even more fascinating from a patient. But being that it's being produced in mammalian cells in the liver, I think that it would be fairly normally modified, and it should be well glycosylated. And we do know that it's taken up and can treat Pompe disease. There's a question from Delphine that's in French. If anyone can help me with translating that, we'll get back to that in a second. I have a question from Dawn in the chat. Do you have a threshold of symptoms, mild or severe, that a patient should have to be able to participate in the gene therapy clinical trial? Yes. I could speak to our study, and both for the 6-minute walk test and the FVC, we had a window where it shouldn't be entirely normal and it shouldn't be too low. And the idea there is, for this clinical trial with just a few participants, to be sure that the participants could both show a benefit. It could, they could improve in terms of their 6-minute walk test, for example, and also that they could still do the test to be able to show the improvement, that their symptoms weren't too severe. So there's like a window for the muscle function test. Now this from Zoom user, if I were interested in participating in a trial, is there one you would suggest? I have to leave that entirely to you, but so you can get information on clinicaltrials.gov. I'd really recommend that because that's a federally sponsored site where all the sponsors who are doing clinical trials have to list their trial and share information about it with the same format, and then talk to your physician, the physician you see about Pompe disease. I think those would be excellent sources of information. But, from my disclosure slide, and, talking about my research, I'm clearly biased in terms of my view. From Kara.
That was a translation from the Delphine.
Oh, okay, great. Yeah, I've been diagnosed for a year. I'm not taking treatment. How can I volunteer for a clinical trial of this gene therapy? So again, clinicaltrials.gov is a source for any clinical trials that have any U.S. involvement, at least. So you can see there, what is the clinical trial? What are the criteria to enroll? In other words, would you be eligible? And what is a contact for that clinical trial? And also where it's happening. There potentially there could just be a site in your country, and you could see that from clinicaltrials.gov. And from Carolyn, are there still limits to participation in gene therapy? I mean, definitely based on the openings for these clinical trials, and I haven't gone into details at all about how many. Tiffany, please go ahead. I saw a note that Tiffany would like to answer the question.
Oh, maybe she's on mute.
Oh, I'm sorry. Yeah, no, maybe we caught her off guard too, but I'll pause for a second.
Sorry, no, I was just saying that you would answer it live.
Oh, I got it. Okay. But yeah. So the way to find out about, if you can enroll is to contact the sponsor, go through clinicaltrials.gov, find the contact information, ask there.
Are y'all able to hear me?
Yes.
Yes, now we can.
Okay, great. Hi. So there's a question that I've asked often, and I'm curious to hear your take on it. We know that Pompe is a spectrum disease, and Ryan alluded to, uptake and the promoters and all of that. But do you think that knowing that ERT isn't, let's say, one size fits all, right? Separate doses are needed. Will gene therapy be one dose for everybody, or would you need alternate doses?
Excellent question. Yeah, and that goes into this question of treating early in life or later. And it's kind of similar for ERT, right? Because, for ERT, higher dosages seem to be needed for treating earlier, and again, at that severe end of the spectrum. For gene therapy, there are multiple factors. So just that the current AAV is gradually lost from cells during growth and development. It's gradually going to be lost, if you treat a patient young over the next years of life, the benefit will be reduced. So different approaches to that include higher doses. From the mouse study I mentioned, we know that can be effective, but not entirely, just going to a higher dose. And then others would be the availability of different drugs for using different AAVs. So at least for laboratory experiments, we can treat with a different AAV and still get a benefit. We could dose once with AAV8, get a benefit, and then later with AAV9 and get another benefit. So you could come up with treating patients multiple times over their lifespan with different gene therapy drugs. And then along that line, go ahead.
Well, I was going to say, I'll let you continue, but redosing, of course, is a huge topic in the field at the moment. So I'd love your thoughts on that as well.
Yeah, absolutely. And with redosing, then there are also strategies just to use a certain really effective immune suppression or depletion of antibodies against the AAV to be able to give the same vector multiple times with benefit. And that really requires further work. It's not ready, not proven in terms of using that in a clinical trial. So I think I'll stop there with that particular question. There's more that could be said. It could be a seminar on its own.
So there's several questions from John.
Mm-hmm.
And the first is, what is the collaboration across universities and other researchers doing with Pompe? How can this be evaluated in research, not in silos?
Yeah. I mean, certainly at the level of the public disclosure of publication of results, even at scientific meetings, where, admittedly, each program is going to give a limited snapshot and update of their research. All that is sharing, and it's really, really important. But the gold standard is publication, review by experts, so-called peer review, and then what's in the scientific journals. And it's highly technical. It requires lots of interpretation, but you can work with your doctors, who are expert in the field, to understand that kind of data. And to me anyway, that peer review and publication, as well as meetings with lots of open sharing wherever possible, are really key in trying to compare the gene therapies. And ultimately the same standards are there in terms of showing benefit, and there will be information to allow comparison. It does take a while, and that's challenging.
Sure. And then another question was, what levels of GAA does the person need to have to be part of the window for treatment?
Can you repeat that or rephrase it a little bit? I didn't quite catch it.
What levels of GAA does a person need to have to be part of the window for treatment?
Levels of GAA?
Yes.
Yeah. Okay. Well, currently, the only gene therapy is in clinical trial, and it'll depend on the criteria for that study. So it's most likely going to be defined both by having a low level of GAA and having, the gene mutations or variants in GAA that confirm the diagnosis of Pompe disease. And it interplays with the age that patients are being enrolled in the study. So for instance, for our study, only greater than 18, and all those patients did have late onset Pompe disease, and for instance, they all had one inactivating variant or mutation, and then the common late onset Pompe disease variant that 90% of patients with late onset Pompe disease have. So I hope that addresses the question somewhat.
Well, I think really what I understood they were getting at is, if you look at inclusion and exclusion criteria,
Right.
has GAA activity been considered? And I don't know that it has.
No, usually just showing that it's deficient, and that'd be based, on the blood GAA, where
Right.
really our laboratory and other laboratories have an affected range, clearly Pompe disease, and then a normal range, and then an intermediate range, where it might be a variant pseudodeficiency, not Pompe. So
it's more just a confirmed diagnosis of Pompe is what you need, not a specific GAA level.
Exactly.
Are there any other questions? All righty then. Well then, oh wait, I'm in my mid-40s with LOPD on Myozyme since 2007. Where should I put my hopes? Gene therapy or more effective ERT?
Okay, I think based on availability, first, more effective ERT. And then depending on the data, and actually, of course, FDA approval, gene therapy. I mean, again, this is just my view, but, I think there could be advantages to gene therapy. It just has to, be fully evaluated and approved.
Is there a intention to go international with your trials or just these international approvals?
Okay, so now this is bordering on sponsor plans, and I'm going to emphasize. Well, I'm at Duke University. I'm a professor, but maybe Ina might from just maybe the stage of development, current developments at AskBio, since you're there. Whatever you'd share.
Sure. Currently at AskBio, our ACTIS 101 study is ongoing. We have patients that are in the long-term follow-up, and we're evaluating our clinical program and the further development of that. And certainly, depending on, I think there is a need to go global, but it really depends on the program and the logistics and things like that. And I'm sure the other sponsors are probably also thinking the same. Typically, phase 1 or part is that your safety study, and depending on the number of patients that you think you need, sponsors work with the regulatory authorities to determine where the sites are. And then, of course, as you move further in your program, where you conduct and you need more patients, that's when they broaden the region that they're doing the clinical trials in.
Thank you for answering that one. And then again, back to you, Dr. Koeberl. Are you currently working with a gene therapy trial?
Right. So the trial at Duke is ongoing, and we're not enrolling right at the moment, but continue to work with the trial with AskBio that I mentioned, that I described.
Thank you. And Don said thank you for answering his question. We also have in the chat a comment from Erica saying, didn't have a question, but wanted to thank you very much that you and your team have given our families so much hope.
Well, you're welcome. Appreciate that comment.
Are there any final questions for Dr. Koeberl or Ina?
Thank you, Tiffany. I'll just say thank you on behalf of AskBio, Tiffany, AMDA, and your team for hosting this webinar. Dr. Koeberl, of course, for your research and for sharing all of that research with us today. We do have a special email address if you'd like to inquire. It's askfirst@askbio.com, askfirst@askbio.com. If you have questions about our trial, otherwise, as Dr. Koeberl mentioned, the clinicaltrials.gov website. And that's it. Thank you, everybody, for joining.